Dear students, I am Dr. Rajni Arora, Associate Professor in Zoology, Swami Shraddhanand College, University of Delhi. I will be speaking on immunology module, that is antigen recognition, where the main focus will be on the subtopic adaptive immunity and antibody. In this module, I will discuss about adaptive immunity, structure of antibody, that is its light chain, heavy chain, hinge region and disulfide bonds. The types of antibodies, that is IgG, IgM, IgA, IgE and IgD. Functions of antibody, that is opsonization, transcytosis, passive immunity and ADCC. After going through this chapter, you all will understand about various aspects of antibody molecule and will be able to answer few questions and deduce important learning outcomes, that is, what is adaptive immunity, why is antibody an important component of adaptive immunity, how it is a component of antigen recognition unit, how do the antibodies function to combat the pathogen, how was the structure of antibody deduced, what are the various isotypes of antibody and what are their functions. Let us now start our topic. As you know that immunity is categorized as innate and adaptive, the adaptive immunity is further classified as humoral and cell-mediated. B and T lymphocytes are the key cells of adaptive immunity. The cells must recognize the immunogen entering the body. Recognition and response are the two properties of the immune cells. B and T cells have the receptors called B cell receptor or BCR and T cell receptor or TCR for recognition. B cells respond by releasing antibody, but T cells form CTL. Adaptive immunity is also called acquired immunity. The person acquires the immunity when attacked by pathogen. It tackles with the intracellular and extracellular antigens by humoral or cell-mediated mechanisms. Humoral immunity is the immunity given by B cells. B cells are activated when they recognize the pathogen. They clonally proliferate and form terminal effector cells called plasma cells, which release antibody in body fluids. Cell-mediated immunity is given by the T cells. It is given in response to attack of intracellular antigen or to tackle the altered cancer cells. T cells on activation also proliferate and finally form CTL, which kill the target cell by certain lytic secretions. Let us study about antibody. The term antibody was coined by V. Behring and Kitasato in 1890. It is both membrane bound and secretory in nature. The membrane bound is located on B cells to recognize the pathogen, but secretory antibody is released in blood and body fluids after B cell activation for various effector functions. But when is the B cell activated? B cell, when encounters the antigen, engulfs and then presents it on MHC1 molecule. Macrophage and dendritic cells can also do the same work, that is act as APC or antigen presenting cell. It is then recognized by T helper cell which releases cytokines. The type of cytokines determine activation of B cell and formation of plasma cells. Plasma cells release antibody. You must know that antibody is an immunoglobulin. It was proved by A. Tiselius and E. A. Kabat that antibody is a gamma globulin. They collected the serum of ova-albumin immunized rabbits and divided into two aliquots. Then mixed the ova-albumin in one of the aliquot and electrophorosed the two serums. Gamma globulin fraction did not appear in aliquot mixed with ova-albumin. What does this wonder molecule, that is antibody, look like? That is its shape and structural detail which make it a component of recognition unit BCR and also able to perform other functions for defense. It is important to know what is BCR. It is the important recognition unit and is made of membrane bound immunoglobulin and disulfide linked heterodimers called Ig alpha and Ig beta polypeptide chains. Do you know antibody has a unique shape? It has a Y-shaped structure and has two light and two heavy chains. The light and the heavy chains have constant and variable domains. Antigen binding is at the variable domain and constant domain is for effector functions. Hinge region provides flexibility to the antibody molecule. How was the structure of antibody discovered? This was a contribution of two scientists, that is G. M. Edelman and R. Porter, for which they got Nobel Prize in 1972. They worked separately using different chemical methods. R. Porter treated the antibody with pepsine and papain and got Fab and Fc or two Fab fragments. Edelman treated the antibody with mercaptoethanol and got four fragments, two of small size called light chains and the other two are called heavy chain. The Y-shaped structure was thus deduced. The antibody is a glycoprotein and the amino acid sequence was studied with the myeloma proteins and the light chain sequence studied using the Benz-Jones proteins. What are the different components of antibody? As discussed earlier, antibody consists of four polypeptide chains, two light and two heavy chains. Each chain has N-terminal variable and a constant domains. Variable domains are VL and VH, whereas the constant are Cl and CH1, CH2, CH3 or sometimes CH4. There are both inter and intra polypeptide disulfide bonds. Antigenic determinants bind to the paratope of variable domains. The hinge region gives flexibility to the antibody molecule. Each monomer can bind to epitopes. It is monoclonal if it binds with one epitope, otherwise polyclonal. Fab fragments are monoclonal. The variable region of antibody is important as it is this region in which most of the differences in antibody fall. This particular variable region of light and heavy chain is called CDR or complementarity determining region. It is the region which binds with antigen for recognition when it is a membrane antibody. After recognition is the activation of B cells and antibody is secreted by the plasma cells for other defense functions. In membrane antibody, there are three components of the carboxyl terminal, that is 26 amino acid hydrophilic spacer sequence, hydrophobic transmembrane sequence and a short cytoplasmic tail. What are the different types of antibodies? There are 5 types of antibodies, that is IgG, M, A, E and IgD. IgM and IgD are the membrane antibodies and are the important component of recognition unit or BCR. Let us study why we have 5 types of antibody. The 5 isotypes of antibody have different properties and functions as studied from the given table. They are named based on the heavy chain type, that is mu, gamma, epsilon, delta and alpha. Accordingly, they are named as IgM, IgG, IgE, IgD, and IgA. IgM is pentameric in secretions and its monomer is also a membrane antibody forming the part of recognition unit. It is the antibody released in primary response, that is, when the body encounters the first antigen. It cannot cross placenta. It is a good agglutinator and complement activator. It has subisotypes with minor functional differences as Ig1, Ig2, Ig3, and Ig4. Let us now study IgG. IgG is a monomer and can bind with the two epitopes. It has 80% concentration in the serum. It is the antibody of secondary response and can cross placenta by transcytosis. It gives passive immunity to fetus, which means fetus gets preformed antibodies. Its Fc component can bind with the phagocytes for ADCC and opsonization. It also neutralizes the toxin. IgA is the antibody of mucosal secretions. It is dimeric in nature. The dimerization requires J chain. It is present in tears, mucosal secretions, and first mother's milk, that is colostrum, again for passive immunity. It takes care of antigens entering the GI tract, respiratory, and urinogenital system. IgE is just 0.002% in the secretions. It is important for allergic and parasitic infections. It has the Fc receptors for basophils and mast cells. It is the cross-linking of the antibody IgE which activates these cells. Binding with these mast cells result in releasing the histamine which cause allergic reactions. It kills large parasites with the secretions. IgD is a membrane antibody. Now about the effector functions of the antibodies. They neutralize the toxin, agglutinate the cells having surface antigens, or precipitate the antigen. They coat the bacteria to be easily phagocytosed by the macrophages. This process is called opsonization. Antibodies also activate the complement system. Antigen and antibody complex activates C1, the first component of classical pathway of complement system. Once it is activated, it kills the bacterial cell by forming a membrane attack complex or MAC. ADCC or antibody dependent cell cytotoxicity is another important function. What actually is ADCC? Antibody coats the target cell. Target cell means the cell which is a pathogen. Natural killer cell recognize the coated target cells. They release lytic enzyme and kill these cells. Epitope, that is antigenic determinant, and paratope is component of antibody binding with the epitope. Interactions of epitope and paratope are non-covalent. If the interaction is with a single epitope and paratope, it is called affinity, but if it is more than one, it is called avidity. Avidity is stronger than affinity. What are the important hotspots in antibody research? They are mainly the production of monoclonal antibody, production of designer antibodies by designing first the antigen from the peptide sequence and preparing the antibody of interest, making different electrochemical sensors. Before I conclude the discussion, let me summarize it and give you the important points of the topic discussed. First, antibody is the important effector molecule of the adaptive immune system. It is a Y-shaped glycoprotein with Fab and Fc components. It has diversity as it is specific to variety of antigens which are encountered in the blood and other body fluids. It is both membrane bound and secretory. Membrane bound antibody is for recognition of antigen and is part of recognition unit called BCR. Each component of antibody, that is light and heavy chain, hinge region, disulfide bonds, J chain and carbohydrate group, gives it a special property and makes it a versatile molecule. It has 5 isotypes as IgA, IgM, IgG, IgD and IgE. Each type functions at strategic locations of the body. The various effector functions like ADCC, complement activation, opsonization, etc. aim to target the antigen and differ in isotypes and sub-isotypes. The antigen-antibody interactions are non-covalent interactions. Thank you for your kind attention and patient listening. Hope you enjoyed the talk.